Hello, everyone. Welcome back to my single-cell proteomics data analysis video tutorials. In the second video, we aggregated the PSM data into peptide data. And you can see in the SCP object, we have 6 assays now, and assay 4, 5, and 6 are peptide data. So in today's video tutorial, we are going to process the peptide data, and later we will aggregate the peptide data into protein data. So far, we analyzed all the assays separately. So now we can join the peptide assays into one assay as the peptide. So we can use the join assays function, and the assays will be 4, 5, and 6, and we give a name as the peptide. So let's join the peptide assays into one peptide. So let's have a look at the object again. You can see now we have 7 assays. Number 7 is the peptide assay. You can see in the peptide assay we have 38 columns. That's the columns added up from 3 assays, 11, 11, and 16. But the rows is not the total number of 3 peptide assays because there are common features among 3 different peptide assays. So now we can plot the SCP again. Let's zoom in. You can see in previous videos we aggregated the PSM data into peptide data. Now we joined the 3 peptide data into one peptide assay. You remember for the sample type in the assays we have blank samples, macrophages, monocyte, and also we have the unused channels, we have the reference channels. For the following analysis, we want to analyze the single cells for macrophages and the monocyte. So we can filter the single cells to keep macrophages and the monocyte. At the moment, we also need the blank channels as negative control to calculate the Q matrix. So we can fill the single cells as blank macrophages and monocyte. So for the Q features data, it is different from the matrix data. In the matrix data, we only have the rows and the columns. But for the Q features data structure, we have the rows, columns, and assays. So the cell types are in the columns, so we can subset the samples. So you can see here, we keep all the rows and all the assays. And in the columns section, we keep cells as blank, macrophages, and monocyte. So let's subset the cells. And we can have a look at the SCP object again. You can see now for each assay, the columns reduced to 8 and 12 in the PSM data and the peptide data, and the columns in the peptide data is 28 now, but the row data was not changed. So next we can filter the single cells based on the medium relative intensity. So first we can calculate the medium relative report iron intensity for each cell. Because we are filtering the single cells in the peptides assay, so we just need to calculate the medium relative intensity for the peptide assay. So we know each column is a single cell. You can see we got the medium relative intensity for each cell. So now we can save the medium relative intensity as SCP column data Then we can use ggplot to create a figure to compare the medium relative intensity for each cell. So let's zoom in to see the medium relative intensity for each cell. You can see here, the blue color is the medium relative intensity for monocyte, and the green color shows the medium relative intensity for macrophages, and the red color shows the negative control, because the negative control channel doesn't have cells. So you can see here, actually the medium relative intensity for both monocyte and macrophages are above the black negative control. So we don't need to filter the single cells according to their value for the medium relative intensity, because this is a demonstration data set. Actually, we have only 28 cells here at the moment. If the sample size increase, then you may have to filter the cells using the medium relative intensity. So we are going to do one more filtration for the peptide data, and we filter the single cells based on the medium CV. So the medium CV parameter measures the consistency of quantification for a group of peptides that belong to the same protein. So for the filtration, we will remove the cells that have high medium CV parameter over the different proteins. So we can calculate the medium CV using the PSEM dataset 1, 2, 3. And we need to use the group by function to provide the protein information. And we only calculate the CV value for a protein that have at least 5 peptides from the dataset. And we will save the medium CV value for each cell as column data in the peptides assay. So let's calculate the medium CV for each cells. Once again, we can use ggplot to create a figure like the medium relative intensity to work out the best value for medium CV to filter the cells. So let's zoom in to see the medium CV value for each cell type. Because the blank negative control channel doesn't have any peptide information, so the medium CV value is quite high, and we can use the medium CV value from the negative control to estimate a value for filtering the cells. And you can see the medium CV value for the macrophages is just below 0.6, and the medium CV value for the monocytes is just over 0.6. So we can set the medium CV value as 0.65 to filter the cells. So let's do the filtration. Use the medium CV value as 0.65. So now we calculated the Q matrix for each single cell, and we don't need the blank channels anymore, so we can remove the blank channels from the SCP dataset. So following the filtration, let's have a look at how many cells left in the SCP peptide dataset. So you can see from the peptides assay, we only have 17 columns. So we only have 17 single cells left in the dataset. So we kept 17 single cells for the rest of the analysis. So next we can process the peptide data. First we can normalize the peptide data. And you already know for the single-cell RNA sequencing data analysis and also the ATAC data analysis or other single-cell protein data analysis, we always normalize the data. And you can use the normalize function from the Q features package to normalize the peptide data, but for the SCP package, they recommend to use the sweep function to normalize the peptide data. To use the sweep function, the column peptide data were first normalized by dividing the relative intensity by the medium relative intensity, and we save the normalized column data as the peptide's normal column. So let's normalize the column data first. Then next we can use the sweep function to normalize the row data. Now we use the peptide's normal column assay and divide the row data by the medium relative intensities, and we create a new assay as the peptide's normal. Because the peptide data always contains missing values, so another common procedure is to remove highly missing data. Here we remove the peptide with more than 99% the missing data, and we can use the filter NA function from the Q features to remove the peptides with the high missing rate. The final step to process the peptide is perform log transformation for the peptide data. We perform log 2 transformation, and we use the assay after peptide normalization and create a new assay as the peptide log transformation. So let's perform the log transformation analysis. Now we can have a look at the SCP data. You can see we created 3 more assays, 8, 9, 10. During the peptide data processing, we normalize the column data, then normalize the row data, then perform the log transformation for the peptide data. We also filter out the high missing rate peptide, and now we only have 367 rows and 17 cells. If we plot the SCP data, let's zoom in. You can see from the plot, we performed the column data normalization, row data normalization, and the log transformation. So now we processed the peptide data. We can aggregate the peptide data into protein data. So for the aggregation, we are going to use the final assay peptide log transformation, and we are going to use the aggregate features function from the SCP package to aggregate the protein data, and we will name a new assay as the proteins. For the protein aggregation, we will aggregate the proteins over the column features needing read the protein. So let's aggregate the peptide into protein data. And we can have a look at the SCP again. You can see now we have a new assay named as the proteins. And in this assay, you can see we have 100 rows and 17 columns. So that means we have 100 proteins in total and 17 cells in total. So let's plot the SCP again. So let's zoom in. You can see now we aggregated the peptide data into protein data. Okay, so I'm going to stop from here for today's demonstration. I hope my video tutorial can help your data analysis. If you haven't subscribed my channel, please click the subscribe button. In my next video tutorial, we are going to process the protein data and perform the PCA map analysis to cluster the cells. Thank you for watching my video tutorials. I hope to see you in my next video tutorial.